Okay, it's my pleasure to introduce Emily Quach. He's our presenter today, and she's also presenting her exit seminar as a master student. Emily came as a transfer in the 2018. She demonstrated grand interest for research. She started working as a volunteer in the aquaponic lab in 2019, and she participated in the SOAR program, the Summer Opportunities for Research in 2021, been working in a project that we collaborate with the USDA, the ARS, with Dr. Milner, and she's gonna present her the results about food safety in aquaponics. Emily.
Thank you for the introduction. So as mentioned before, my name is Emily Quach, and my research is on the survival of E. coli and changes in physicochemical parameters in aquaponic systems during basil and lettuce production. So just like an overview about what my presentation is about and kind of the things I'm going to be talking about today. First, I'm going to be introducing aquaponics, the technology behind it, the benefits and the challenges that come across with this technology, my hypothesis and objectives for the overall study. I did 2 experiments for my master's research, so I'll be going through the first one, so the methodology and implementation and the results for that, as well as the methodology and results for the second one, and then concluding in the grand scheme of what this research means in the field. So first of all, what is aquaponics? It's a type of agriculture food production method that integrates aquaculture, which is fish farming, and hydroponics, which is a form of soilless agriculture. In this case, it uses fish waste as a form of fertilizer to provide nutrients for plants. In this diagram here shows a deep well water aquaponic system, which is the type of aquaponic system that I am working on, working with for my research. And at the bottom, here is the fish tank. So common fish species are tilapia, for example. The fish waste is then pumped up. So typically in the fish tank, there is obviously a water pump. There's also an air pump for proper oxygen levels for the fish species that you're working with. The water pump then pumps it through a filter, which takes out solids. And then that is then pumped up to the upper grow bed, where it's actually submerged in all water, and a raft is used to elevate the upper parts of the plant, so only the roots are submerged in the water. And then the discharge gets flown down back into the fish tank. So what are the benefits of aquaponics? Well, it can address concerns like urban population growth, farmland shortage, greenhouse gas emissions associated with food transportation, and resource insecurity. This is because this type of technology can be pretty much built anywhere indoors, as long as you have access to a stable energy grid, as well as a consistent source of fresh water. So because of that, it can be grown pretty close to urban centers. And so transportation is drastically decreased as a result. So which then contributes to less greenhouse gas emissions when you transport it across shorter distances. Some challenges that are associated with this type of technology is energy costs. These systems do have to be run on electricity 24 hours a day, 7 days a week, because the water pumps, the LED lights, air pumps, everything has to be run monitoring temperature indoors, requires electricity. And as a result, it may not be as sustainable depending on where your energy comes from. If it's produced from coal or petroleum, for example, then it's not as sustainable as a location where it has more solar power or wind energy or hydropower, for example. There's also high startup costs. You do have to purchase all of these materials beforehand before you can even start your production system or your operation. And so that's a pretty high investment right off the bat if you are hesitant about starting this type of technology. So you do have to make a pretty large commitment before starting. And then there's potential food safety hazards. This is a huge research gap. There really isn't that much research available as of right now that we can access that even really talks about food safety hazards for aquaponics. And so because of that, there's a lot of hesitancy about pushing this into the commercial space just yet, because we just don't really know what happens if something like E. coli does happen. Like what can we do to mitigate it, or what can we do to prevent it, or even clean up, for example. So that goes into my research. So I hypothesized that in healthy aquaponic systems, E. coli will not survive past 14 days under controlled condition. This is based on a paper that suggested keeping aquaponic systems alone for 8 to 16 days can drastically reduce the risk. So I just chose 14 days as a 2 weeks within that range of 8 to 16 days like that paper suggested. That's possibly a chance to indicate that there is no E. coli, and so the risk is very low or not there. So that goes into my objectives. The first objective is to determine the survival of E. coli in our aquaponic research units. The second objective is to determine E. coli survival on leaves and roots of basil and lettuce, which is our selected plants for our study. And the third objective is to evaluate the association of physicochemical water quality parameters. So going to my first experiment, it's the preliminary phase. Before we start on working with aquaponic systems, we wanted to know what would happen just in using the aquaponic effluent. We didn't want to just start inoculating on these systems without really knowing what kind of behavior or change in population we would expect within the first few days. So we started with a bottle lab scale study, a bench scale study actually, where we got our effluent from a mature aquaponic system that is currently running in our lab. That's just for teaching instructional use only. And we have 4 treatment groups and 1 control group. Starting on the very left, in the white, A is our first treatment group, which is just distilled water, and it's inoculated with E. coli. Our second group, B, is a mixture of aquaponic effluent with distilled water, so it's a 33% concentration of aquaponic effluent that's also inoculated with E. coli. Our next group is 100% aquaponic effluent that is inoculated with E. coli, and our control group is 100% aquaponic effluent that is not inoculated at all. And our final group is a 100% aquaponic effluent concentration that is inoculated with E. coli. The only difference is that each day a certain volume, in this case 105 milliliters, was taken out and replenished with fresh aquaponic effluent to simulate a nutrient replenishment. All of that was placed onto a shaker kept at room temperature at 150 RPMs, and our intended duration of the study was 14 days, but we didn't really get results after a few days, so we actually didn't do the full 14 days. So the analysis that we were doing was microbial enumeration analysis as well as physicochemical parameter analysis. So at the very left, figure 3 is the mature aquaponic system that we used. We in the lab call it the IBC system just in general. So if you do see IBC in the graphs later on, I'm just referring to the aquaponic effluent that is taken from this system. Figure 4 shows the setup for the preliminary bench scale study. The left, the incubator is used for the microbial enumeration analysis. And the setup on the right has the shaker as well as a container that's holding the bottles that has the different mixtures of effluent in it. And then figure 5 shows a configuration for all the treatment groups and control groups. So for microbial enumeration analysis, we were looking at aerobic population, which is just a natural population of bacteria that is there, as well as E. coli. So aerobic bacteria was analyzed using APC Petri film. The aerobic bacteria does include E. coli because it is an aerobic bacteria, so that will be taken into account in the population in addition to the natural microbial community. E. coli was monitored through spread planing. Specifically, we use McConkey agar rifampicin. Our E. coli strain that we are using is called E. coli TVS 354, which is a non-pathogenic strain of E. coli that is rifampicin resistant. Rifampicin is a type of antibiotic. So that allows us to be able to just count the colonies for our specific strain of E. coli. So you can see figure 6 is the agar spread plating that we used, and we just count the red dots for every colony. And then figure 7 is a picture of Petri film. We count the pink dots. It's kind of hard to see because they're very small. It can be a very tedious process to sit there and count all of them, but that is a part of it. And for physicochemical analysis, we looked at pH, temperature, electroconductivity, ammonia, and nitrate. So looking at the results for the first experiment, starting on the very left of figure 8, This has the 3 treatment groups of distilled water, 33% aquaponic effluent and 100% aquaponic effluent. And we saw there was a drastic decrease in the first 24 hours, but it did take longer for the 100% aquaponic effluent to decrease. But by 72 hours, it reached undetectable levels through spread plating, which is why we did not continue past day 3. It wouldn't have made sense because it just would have been 0 for the remainder of the 14 days. Looking at figure 9, because we were taking out a considerable volume of aquaponic effluent, we did a different type of test, such as called a quantitrate, to look at the E. coli population. And we saw that there was a less drastic decrease over the course of the 72 hours. And by 72 hours, there was actually still some population of E. coli left. In terms of the aerobic bacteria, which is colored in blue, there was an increase in the first 24 hours, but then a decrease by 72 hours. That trend is very similar to the 33% aquaponic effluent group and the 100% aquaponic effluent group as well. It has a very similar pattern. Distilled water had an increase and then a decrease, but distilled water was just showing the E. coli population. And the green in this case is in figure 10 is actually showing the uninoculated control group. So it has a very similar pattern compared to its inoculated counterparts. So going into the second experiment, which is our main experiment, we're actually inoculating into functioning aquaponic systems. We have a total of 12 deep water aquaponic systems, 2 treatment groups and 1 control group. So there was 4 replicates of each. And our control group was just left as is. We didn't do anything to it. No introduction of any type of bacteria. For the first treatment group, we inoculated in a low dose concentration. So it's at 2 log colony forming units per milliliter. And for a second treatment group, it was a high dose inoculation of 4 log colony forming units per milliliter. All the systems were running for 4 weeks at room temperature. So similar parameters and conditions in the previous experiment as well. And the same colony enumeration analysis and physicochemical parameters were analyzed. Each system had 2 basil plants and 2 lettuce plants. Because these systems are small, we can't really add any more plants. We did want to see the difference between how the 2 different types of plants would have different populations, or if there were any at all. So in the implementation, figure 12 shows a diagram of the systems that I was working with. So starting with figure 1, number 1, it is the fish tank. It had an air pump tube connected to it, as well as a water tank and the water pump. The water pump would pump the water up to our filtration system that not only took out physical solids, but also provided ample space for a lot of surface area for bacteria to grow on, because there are also nitrifying bacteria that convert the ammonia into forms of nitrogen that plants can use. A discharge is flown into number 2, which is our deep water system that holds the 4 plants. So only the roots are submerged in the water. And then that excess discharge is then flown back down into tank 1 through number 4 and 5. Figure 13 shows a picture of just that discharge system from the top of the grow bed down to the fish tank. And we're able to control the water level just by the tubing here. So if we wanted to be raised higher, we can. If there's more root structures, we can increase the water level. And then figure 14 is the configuration for the plants. So we had Italian lettuce on the left and little gem lettuce on the right, 2 plants each in the system. So we conducted 2 trials for this experiment. The main differences between the 2 trials is that our low dose inoculation was different and our collection points were different. So in the first trial, we had a low dose inoculation of 3 log colony forming units per milliliter. So it's slightly higher. And our collection point was only the water from the fish tank. In the second trial, we had a low dose inoculation of 2 log colony forming units per milliliter. And our collection points were, in the beginning, it was the fish tank, then we transitioned into the filter in hopes of a higher population count that lasted a longer period of time. So looking at the results of the first trial, in figure 15, it's the changes in aerobic bacteria population. So we see that it's pretty leveled and stays consistently the same up until day 10, and then there's an increase and then decrease. We're not exactly, we could be due to a different variety of factors, but that's something we would like to expand on. What is that relationship in the aerobic community, the native aerobic community itself, and how it changes over the course of time. Looking at figure 16 is the E. coli population. It became undetectable after the first day. So we didn't really have any counts for the remainder of the duration of the experiment. So we decided to redo it in our second trial, seeing if there was a difference in population. And in the aerobic bacteria population, looking in the filter, it was kept pretty leveled and there wasn't really a significant change in the population. However, looking at figure 18 and 19, we see that the change in E. coli population had a downwards trend. The first 2 days was collected in the tank water, but we realized that by the third day, it would have been undetectable. So we shifted on the second day to the filters, which is what you see in figure 19. See that the concentration is around the same, but by the next week, it was also undetectable. So we decided to confirm our results through a process of enrichment, just to give us a plus or minus population presence. Throughout the remainder of the experiment, E. coli cells were still present. It was just undetectable through spread plating. So looking at physicochemical parameters, starting in figure 20, we struggled with pH constantly decreasing throughout the first trial, which is why you see a downwards pattern. It could be due to a variety of other factors, but this was a pattern that we saw in our mature aquaponic systems as well. So in our second trial, we increased the pH using potassium hydroxide. So that's why you see it's an upward trend. We tried to keep it around 6 to 7, ideally not as fluctuating as much. In terms of electroconductivity, there really wasn't a pattern that we could pinpoint at this time. We're still doing additional statistical analysis to see if there is any trend that we can see, but it does increase in the first trend, but in the second one, there really is no pattern that we can pinpoint. Looking at ammonia, in the first trial, we provided the fish with more food, so there was more waste that was generated, which contributed to higher ammonia levels than we would have liked. So in the second trial, we decreased the feed, so there was less waste. That's why you see a decrease after the first few days. Because of nitrifying bacteria, they have a relationship of converting ammonia to nitrite and then nitrate, which is a form that plants then can use. You see a similar pattern on the first day for nitrite. In figure 26, you see this increase in the first trial, and then you see this lower level. However, at the end, it has a very contrasting concentrations that we would like to look further into in terms of what may have caused this. Looking at nitrate as well, there was an increase in the first trial, but during the second trial, it was fluctuating a lot. So this is something that we would still like to research further on and look into specifically what may have caused this and what kind of relationship it may have with other parameters. So looking at the plant results, after day 30, which was 4 weeks into the experiment, we harvested the plants. We did the same microbial enumeration analysis, which was spread plating and Petri film. So figure 30 is the aerobic bacteria population, so we use Petri film for that. And we saw that the low dose had the highest concentration of bacteria, highest population of bacteria, and the uninoculated and high dose had pretty similar ones in the leaves, which is on the left side of that graph. The roots in general had lower aerobic bacteria population than the leaves, which could be due to the fact that the leaves are exposed to more oxygen and they're coming less in contact with the water, so that might provide a more ample environment, but we would like to expand more on this through further research. No E. coli colonies were detected through spread plating, but we are able to detect it through enrichment only in the high dose, so the low dose did not have any E. coli cells present. Looking at the lettuce leaves, the lettuce population, starting in figure 31, the lettuce leaf in aerobic bacteria population. We also see similarly that the low dose had a higher aerobic bacteria population, and the uninoculated high dose had pretty similar ones. That being said, the population for the lettuce leaves is higher than the basil leaves. This could be due to because the lettuce leaves are shorter, so the leaves at the bottom are coming in contact with water more often, so they're coming in contact with the native bacteria a lot more frequently, but also getting exposure to a lot of air. The roots overall had a lower bacteria population, but the uninoculated roots had a much higher population than the other two. And looking at E. coli, only the high dose treatment group was detected through spread plating, but we did confirm that E. coli cells were present in the low dose. It was just not seen through spread plating. In terms of the lettuce roots, the dark blue is the uninoculated group, so we did find E. coli there. So contamination did occur at some point, despite being precautious and sanitizing between each samples. We still had contamination occur at some point, which could indicate a risk that human contact can have in these types of systems. The roots had a higher concentration of E. coli population compared to the high dose, which was interesting. It's a reverse of the lettuce leaves. But looking at my hypothesis, as I mentioned earlier, we hypothesized that it would not survive past 14 days, which clearly was not the case, verified through enrichment, but also seen in the plants. They did indeed survive. So that provides a lot of new insight on the survival of E. coli in different parts of the aquaponic systems. And hopefully we can use this research as well as further research studies in the future to be able to develop recommendations for food safety guidelines to mitigate risks in aquaponic systems, specifically in our case, leafy greens, since that's what we worked with. And hopefully we can expand on future food safety research in the future, such as working on listeria and salmonella, as they are also concerns in food outbreaks as well. I wanted to thank not only my committee, as Dr. Yankos, Dr. Zursa, and Dr. Milner. I also wanted to thank Dr. BJ Chetri and Alexis DeBleeker working at the USDA, helping process some of the samples and collecting things, as well as allowing us to use some of their equipment for analysis. And I also want to thank Madeline Doherty and Jillian Ziegler for helping me collect the data and doing all of the laborious spread plating with me throughout the entire study. I'm going to open the floor to any questions.
Great, thank you.
Yes.
You didn't talk about it, but I was wondering if you could say a little bit about like the filtration process and what you were seeing in terms of like total suspended solids with the biofilter. Is it like, did you do any measurements of that? You just didn't report on it, or do you have some anecdotal?
We didn't do any specific analysis on total solids in the filters, but there was a lot of fish waste. We have like 3 different layers in our filters. The top one is just to filter out solids. Because we saw a lot of solids from fish waste. That was why we had to decrease it. So in the second trial, we saw a lot less, but we didn't do any specific analysis on total suspended solids. Sorry.
I mean, what's the water look like in terms of turbidity?
It's clear. You can, the picture's not that great, but it is clear. It's just a lot yellow. But in the rest of the system, there isn't that much like solids that were noticeable. Yes.
I think earlier you said that a previous study had shown most E. coli were lasting between 8 to 16 days, and then in your first experiment, you really weren't finding any levels past 3 days. Why don't you think they were surviving very long, or as long as you expected?
So I do want to clarify on that statement, not that they weren't surviving past 8 to 16 days. The paper suggested that risks were drastically decreased within the range of 8 to 16 days, but may or may not completely be like uninhabitable or not survive at all. In our case, we think it might be due to that the lack of surface area in free floating water. There really isn't much surfaces for the bacteria to attach to and grow, and as a result, they're like competing in a free flowing environment. So that could be a reason as to why, but it also could be because of competition with microbial communities that are already present. But yeah.
Was there a reason you didn't do statistical analysis? But you have replication here.
We're still doing statistical analysis. We just haven't really finished it. So, yes.
I had a question about the pH. So the pH values seem, pretty drastic. That's a pretty big drop that you saw. And so I wondered, do they normally buffer these aquaponic systems somehow? Is that, or what, I guess, or if not, why?
Buffering does occur regularly. It's just it just varies depending on the farmer and the decision to buffer it. And in terms of what they use to buffer it, but it is still a pretty common thing to do. In terms of safety, I was hesitant to do it because I didn't want it to impact I kind of wanted to leave it alone as much as possible, but it did reach such low levels that we had to buffer it. But yeah.
I cannot. It's quite common to use calcium carbonate or potassium carbonate, which also not only increases the pH but helps with micronutrients. With water.
Okay, that was, yeah, I was wondering about well, this, and I guess the stress to the fish as well, or?
Not really.
Oh, not really, okay.
Well,
Interesting.
actually, I have a question that might relate to that. So you would assume in a setup like this, you want it to get pretty stable. But your ammonia levels climb very fast, which probably suggests that wasn't happening. Were these sort of set up to start your trial, but they hadn't had multiple weeks to establish microbial communities and come into balance?
The fish were actually left there for like many weeks without any plants in it. And it was pretty stable, but it wasn't until we started the experiment that it started to decrease increase and decrease in ammonia. Part of it, I think, is due to the fact that we did increase the feed and increase the frequency of the feed once we started the experiment, because we wanted to provide ample nutrients for the plants that may have contributed to the increased ammonia.
So how much do the fish grow in this period of time?
We didn't weigh the fish, but because we were limiting them, they were growing fairly slowly from a visual perspective.
Okay. Other questions? All right, well then we'll thank Emily. Thank
All right,
you.
thank you so much.
There is an email link for your evaluation, so we'd love to get some feedback on the presentation today. It is shared online as well with the audience. All right.
Do you want me to?
No, I can handle it. All right, thank you.
Thank you.
Nice job.
Thank you.